The broadcast is now starting. All attendees are in listen-only mode.
Well, hello, everyone. My name is Güliz Barkan, and I am the president of the American Society of Cytopathology. I would like to welcome you to this special brought to you by the American Society of Cytopathology. This session, How to Critique a Paper, kickstarts the eJournal committee sessions, and this particular session is actually a collaboration of the eJournal and the product innovation committees. Now, this session is very near and dear to me, and I will tell you why. As you know, my dear colleague and mentor, Dr. Stefan Pambucyan, was the chair of the eJournal committee. And I had asked him to do this session, which he had gladly accepted. For those of you who had the pleasure of working and interacting with him, you know what an amazing individual Dr. Pambucyan was. Aside from being a diagnostic genius, he was a very generous human being, and he was very generous with his time, and he was an extraordinary educator. He read 5 to 10 papers a day, and he liked papers, so he would print them out, and he would put them in his jacket, and then, go home on a very late hour and read all those papers at the end of the night. And he read the papers in a critical way. He would dig deep into the papers and understand the essence of them. He would say, you should be able to describe the aim, research methods, and results with such clarity that your mother, father, spouse would understand. And journal clubs were fun with him. He would ask questions that would make me think, or anybody think, this is brilliant. I didn't even think of this. Like, that's such a critical way of looking at it. So at Loyola, he started doing a session on how to critique a paper, how to run a journal club, to set the tone of the journal clubs during the year. So for this reason, this session is dedicated to our own Dr. Stefan Pambucyan. Now, I would also like to thank Dr. Rezaei for stepping up and taking on as the chair of the eJournal committee. I also would like to thank our panelists for helping put this session together so quickly. And just as a little side note, as Dr. Pambucyan would say, Dr. Thrall was a former resident of Dr. Pambucyan at the University of Minnesota. And I must say, Dr. Pambucyan spoke very highly of you all the time and how eloquent you were with writing papers. Dr. Weiss is our first Shark Tank winner, and she's the vice chair of the Product Innovation Committee, and Dr. VanderLaan, as you know, is the associate editor of JASC. Critically reading papers is not important to understand and further our work, but it also helps better our own writing skills. Furthermore, when we become reviewers and hence gatekeepers of information, this skill becomes invaluable as a reviewer. I hope you will all enjoy this session. There you have it.
Thank you very much, Dr. Barkan, for the nice introduction to our first interactive eJournal series, actually, for 2021. I am very humbled to be the chair of this committee, and I am very grateful for our gracious panelists who accepted to work with us in a very short time, as you said, to put together this first eJournal series. There is no conflict of interest. In the interest of time, I'm not going to read every single item on the slides for the introduction part. They have very impressive bios. Many of you probably know them already. But in alphabetic orders, I just put the very few highlights of what they have done as their impressive work, and as the service to the community of the scientific community, especially in the cytology community. Dr. Thrall is a professor of pathology and genomic medicine in Houston Methodist Institute of Academic Medicine, and also a professor of clinical pathology and laboratory medicine at Weill Cornell College of Medicine. He is the cytopathology fellowship director and director of digital pathology at Houston Methodist Hospital. And he is member of many of the ASC committees, including the ASC Scientific Program Committee and chair of the ASC Small Biopsy Task Force. He is member of editorial boards of JASC and Archives of Pathology and co-editor of Diagnostic Pathology. He's author and co-author of 55 peer-reviewed journal articles. Our next panelist, Dr. VanderLaan, he is an associate professor of pathology at Harvard Medical School and director of cytopathology and thoracic pathology at the Thrall Medical Center. He is the associate editor of the JASC and member of editorial boards for 5 other pathology and medical journals. And he has published over 130 original research papers, review articles, editorials, and book chapters. Our next panelist is Dr. Vivian Weiss. She's an assistant professor of pathology and microbiology and immunology at Vanderbilt University. She is the vice chair of the Laboratory Service Committee of the American Thyroid Association, as well as the Product Innovation Committee of the ASC. And she is an editorial board member of the Cancer Cytopathology. And she also is a principal investigator of an independently funded basic science and translational research laboratory. She has published 33 original research papers, review articles, editorials, and book chapters. With that, I think you know how grateful I am for them to be the panelists on this talk, because what we are going to do is talking about how to critiquing a paper. Before doing that, I think just to engage the audience and to know a little bit about the background of you, and also to set the tone for the rest of the discussion, I would like for you to take the quick poll. Which of the following best describes you? And you have the choices of cytopathologist, pathologist, cytotechnologist, resident, fellow, and cytotechnology students. I think we can click the poll. Okay, we have a nice mix, and about 30-30 cytopathologists, pathologists, cytotechnologists. We have lower numbers of residents, fellows, and cytotechnology students, but hopefully they get what they need to get out of it. The next question, which of the following, oh, no, that's not the next question. Sorry, you already did that. The next one, somehow I'm not able to move. Okay. Do you have a journal club? Okay, journal club or a session that published papers are reviewed at your institution? Yes or no? And okay. And I think we can see the poll. All right, 50-50. That's a very round numbers. All right, the next one we're going to talk about is going to be, okay. This actually is very important. So when reading an article, what section do you think is most likely to be skipped over or just only glanced at? Introduction, material and method, results, or discussion? Okay, let's see. Okay, as expected, materials and methods is 2/3 of the audience felt like that would be where they kind of either skip or glance over. Very good. All right, so I guess now we can go back to where we want to take our, oh, sorry, that's not what we wanted to do. Okay. So before I open the discussion to our super panelists, I kind of would like to kind of go over for the critiquing of the paper. It would be great to have an idea how to go through the different sections of an article when you're reviewing the article. So let's see if we can start with, I believe what I had in mind, it was that to ask our panelists, and I'm going to start with Dr. Weiss, Vivian. In your opinion, what makes a solid introduction? What is the purpose? And what are the things that should be a must-have elements of it? How long and how detailed should it be? Thank you.
Great question. I think the most important part of an introduction is to clearly and somewhat succinctly state the problem that your research is addressing. it is really important for the reader to know exactly what the issue is and what the problem is up front as soon as they start reading the paper. And I like to say, and I think this is on the slide in front of you, I like to say that this is, the first impression of the paper. So when you meet someone, you want to put a good first impression. When you start your introduction, you need to captivate your audience. You need to explain the problem and really get everyone who's reading it excited to continue even reading the materials and methods. And the key to that really is to make it interesting and engaging and to tell a story. And I typically start with, what is the critical problem in your field that you are addressing? And then after that, I like to define the background that has led up to your research. So put it in the setting. What is going on in the research world up until your fantastic finding? And then I think the other thing, which I sort of touched on before, is why is this important? So not only what problem are you addressing, but what is the relevance to the field? Why should people continue reading and continue caring about your project? If you write too long of an introduction, people get lost, right? If it's too short, maybe you don't convey everything you need to convey. And so the sentences need to be clear and engaging and clearly outline the problem, the background, and why this is relevant.
Very good. That was very nicely put. Now let's turn to Mike, because the majority of our audience had skipped over materials and methods. So we kind of thought that would be the answer. It's probably the least intuitive section for many readers. As a writer and as a reviewer, tell us about the importance of this section and what are the key elements to pay particular attention to.
Okay, so materials and methods, I can start out with just what I was told a long time ago. I think this was in a science college course, even before medical school. The point of the materials and methods really is to make it so that if somebody else wanted to repeat what you did in their own institution or in their own lab, they would be able to do so just by copying what you've described in your materials and methods. That's the basic idea behind it. I think a lot of people, they don't even have a basic idea of what's supposed to go in there. But once you understand that basic idea, that what you're trying to do is allow other people to follow in your footsteps and redo what you've done. And not that anyone really ever does that very often, but that's a theoretical idea, at least, is that you're trying to give people enough information so they can really understand in detail what you've done, and they could actually copy what you've done exactly if they wanted to. And really, if you have this concept in mind, it helps you to get to the main point of this, which, as a practical matter, people won't actually try to reproduce you entirely, but they do want to have that information so that they can compare what you've done with what other people have done, and to compare what you have done with what they do in their own lab. But even if they're not doing a study, if you're talking about a new immuno, or if you're talking about a new diagnostic terminology system, they like to say, well, how does what they do in their lab compare to what I do in my lab? And can I take what they've done and expect to get similar results in my circumstances? And it also is useful if you have all that detail, so that if somebody's doing some sort of a review of different papers that maybe have different findings that seem on the surface similar, they can go into the materials and methods section and figure out, okay, well, why did these different authors in different institutions get different results? Is there a real problem here, or is it just that they were using different methods and maybe the results do make sense after all? So I think that these are the basic ideas you need to keep in mind when you're writing this, and when you're reviewing or reading this, you should be looking to have something that has enough information so that you feel like you really understand what the person did and how you could yourself apply what they've done or compare it to other papers that are similar.
That's very good. Go ahead. Sorry.
So, and the other things to, the basic things to include then are, the population you're studying, the laboratory methods, what equipment you're using to do what you've done, the statistics and the methods of statistics that you're using, and some sort of statement about your IRB approval is usually in there as well. Mm-hmm. So those are the basic things to cover. And I think that, it really, The materials and methods oftentimes is the hardest to write because you're supposed to refrain from editorializing. You're supposed to just describe things. And that's, again, where sometimes authors fall down because they feel like they are kind of unsure what to say. But if you just try to make it with the mindset that you're going to help someone else copy your study, that's the basic idea.
Very good. That was very nicely put. Thank you very much.
If you don't mind, I'll jump in here, get a little discussion going here. I think the materials and methods section is so important for all of those reasons that Mike just outlined. And two things, I'd like to kind of touch on. First off, I had the pleasure of being involved in a CAP guideline, creation. And one of the things that major guidelines or meta-analyses or reviews really rely on is being able to dive into the materials and methods to know exactly what was done. And it's really unfortunate that so many papers were excluded from the systematic review, from inclusion in these guideline statements because of the lack of any details, adequate details in the materials and methods. And so it's critically important to know what you did, how you did it, and how the results of the paper really are based on what was done. As Mike said, you can have two, similarly constructed studies that have very divergent results. And it doesn't mean that either one is wrong, but there might be, again, reasons in just in the way the assays were technically performed. And, Vivian, I'm very curious to hear your thoughts on, again, in the lab. I remember as a grad student, you eagerly look for, if you're going to set up a new assay, you're going to do something, it's the materials and methods that you want to be able to reproduce exactly, what other labs did.
Yes, Paul, that's exactly right. And I think Mike said this as well. So often we'll read a paper. I had a grad student come to me the other day and say, hey, I want to do this single cell sequencing on formalin fixed paraffin embedded tissue while retaining the architecture. And there's this paper that was just published and I'm going to do it based on their methods. And you have to remind them, the methods aren't always written in such a way that the study can be reproduced. They usually just kind of summarize briefly the tools that they use in the equipment. And often they're not written in a way that someone can come after and do the same study in exactly the same way. And that actually has caused a lot of issues in science. There's a lot of lack of rigor and reproducibility because of the lack of attention to the methods section. So I think we can all do each other a great service by clarifying those materials and methods so that someone isn't having to repeat all of that after you and reinvent the wheel after you.
And another kind of point along those lines is, kind of putting on a, associated editor hat. Sure, it would be great to think about every paper that we put out into the world, someone reads, cover to cover, every single word. But that's just not the reality. Some people are looking for more, some people are looking for less. And so I want to really put in a plug now that so many journals are, online and there are so many supplemental material options. I think that it's totally fine to trim down for the actual published paper your materials and methods to just the essentials so people have an idea of what you did. But then if you have a really comprehensive, materials and methods section online as a supplemental material, that's fantastic because people who are interested or who need to know that information can find it. But then again, you're not blowing, 80% of the, words allotted to your manuscript to, describing every antibody clone, every incubation time, every, your cell block method, exactly how everything is done. Super important stuff. But hey, my real encouragement would be to really utilize that supplemental online material that a lot of journals have.
Perfect. I think we did very good justice to the materials and methods because as we all saw and we expected, that was the area that needed a lot more clarification and discussion. So Paul, don't put your hats down yet. As the associate editor, I would like for you to tell us what should be presented in the results section. Is there a most appropriate format for conveying the results?
Oh boy, where to begin? So I'm gonna work backwards a little bit. And again, this is something that I'm really passionate about. So in the results section, as Mike really alluded to, you're really not editorializing yet. You're presenting the results of the experiments of the study. Just here's the data. And, we'll get into the statistical methods and how the data is presented. But how the data is presented is incredibly important in my mind. Honestly, nothing is more soul-crushing than getting a paper that has 7 tables. No figures, no charts, 7 tables. And honestly, tables are super important, and it's a very efficient way of presenting data and large amounts of data. But they're boring, and a table is not always the most effective use of or the best way to present that data. So really think about, how can you present data? Is a pie chart really a good way to show a distribution? If you're looking at a distribution of data, box and whisker plot or violin plots are super helpful, again, to get an understanding for that just a mean or a median wouldn't give you. If you're looking at trends over time, stacked bars charts are incredibly important. So, this is something that I've really tried to do in my studies, and something that I really do try to encourage in papers that I review, and for recommending, suggesting acceptance in journals, is to have interesting, creative, well thought out ways to present your data. And as just a little plug, I went to my library shelf. I don't know if so Edward Tufte. I don't know. So there are these great books. So the visual display of quantitative information, visual explanations, envisioning information. There really is a science behind how data is presented, and really it's an eye-opening way to to reevaluate how you construct charts, what you put into charts, and what you want the reader to take from that. And so with that, Vivian, Mike, any thoughts on the results? Like, what are you looking for? What should be there? And what's the best way to convey that information?
I would second what you're saying, that any articles that take off and get a lot of attention, they usually have at least one sort of killer graphic in them that people can put into a PowerPoint and quickly illustrate the main thrust of the article and what the authors found. So I think that I think I can't emphasize enough. I mean, I agree 100% with what you said, that spending more time thinking and less time writing is the best way to get this. What you want is something succinct. But in order to make something that's succinct and really gets across what you're trying to say, and it really impresses people, there's a lot of thought beforehand, and then just something, very elegant in the execution. And so I think that that's really key for maybe the results section more than any of the others, because really the results are the real meat of the paper. I mean, people tend to focus on the discussion, and I know I tend to do that too because I like to wax eloquent, but really the results are what you're actually contributing to science. The discussion a lot of times kind of meanders off and may not, of course you need it and it's important, but the results are what everything is ultimately based on.
If I may just jump in, Paul, there is a question that is asked from you if you can share the books, like the titles of the books. We can. Yeah,
absolutely. I'll either if there's a lull in the conversation, I'll either throw them into the chat, or afterwards I'm sure we can
sure.
Put it somewhere out there. But I will absolutely share these, and again, they're very eye opening to me, and so y'all might find them useful as well. But yeah, absolutely, I will pass those along. Edward Tufte, Tufte, is the last guy's last name.
All right, very good, very good.
And I would just add on to that and emphasize, the results and the figures that you present are the meat of your study. They really are the most important thing. And I think as a panel, we can't stress that enough. You need to present it in a digestible fashion, and 7 tables is just not digestible. It needs to be something that the reader can look at and understand what you're doing and the results that you're providing with your study. And if you look at it and say, or show it to your mom or your friend, or, and say, hey, can you understand this? Because on first glance, it needs to be readable and understandable, even to people outside of your field. Very true.
Very true. So I'm going to ask one more question from Paul, and everybody else, please jump in. is there any golden rules of understanding the statistical data? And it would be something very brief that you can tell. You can even suggest some readings for that, because I personally don't think it has been, at least not for me, part of the curriculum as I was, being a resident in pathology and doing my fellowship and everything else. So I kind of struggled with those, and I don't know if others feel the same, but I think it would be a good way to just give a punchline for us and then point us to where we can get those information.
Yeah, great. Another easy softball question. Thank you for that. Statistics. Obviously you have to learn a lot on the job, and I'm very jealous of my colleagues that have an MPH or who have done a really in-depth biostats courses. So I guess it's a little glib, but I would say find a friend or find a collaborator or someone who's really good at stats, and buy them beer, buy them chocolates, and that will pay dividends 100-fold. With that being said, again, I'm kind of working backwards. A lot of institutions, a lot of hospitals, a lot of universities have core facilities, and at Harvard there's a biostatistics core facility. So,
right, if
you're doing something more than simple t-tests, Fisher exact test, chi-squared, kappa correlations, where you can do that in Excel or on simple, statistical programs, you might need to have a consultation with a statistician. And increasingly so on journals, one of the questions and one of the things that we have to consider is whether this paper warrants a statistical evaluation, actually have a statistician go over the data set and the methods that were used to compare the statistics. So as far as resources, that's a great question. And I do want to bring everyone's attention to a couple of things. I'm sure many in our audience knows about this. So the first is in JAMA. In JAMA, over the last 3 years or so, they've had an excellent series. So every couple of weeks, they have another editorial or review article on basically their guide to statistics in medicine section. And I think that there's probably about 20 of them that have been published thus far. And it's really a great way to focus in on a certain population or statistical method that's the topic of each one of those papers. And so JAMA, the Guide to Statistics in Medicine. And then again, from my wheelhouse, the Journal of Thoracic Oncology has also in 2020 instituted a similar series, and so statistics in oncology research. And so if you look, and I believe the New England Journal also has some of them as well. Obviously, these are usually more population-based studies, but you can get a lot of great information from the big journals because of a recognition that this is important. So those are the two things I would throw out there. But again, JAMA, the JAMA series is something I would suggest to everyone. But yeah, Vivian, Mike, I don't know if you have any other wisdom, if you are blessed with statistical brilliance, or if you like me, you just find people to bribe and bring into your circle of research.
We usually collaborate with a statistician, and I have a couple of statisticians that are wonderful that we work with extensively. If we're doing a simple t-test or something, we certainly can do it. We actually even do some more complex statistics in my lab, but those are all at the guidance of a really strong statistical team because we do so much sequencing and computation. I am not going to be an expert in all of those statistical tests. So it's really great. And most institutions, as you said, Paul, have resources that you may not even know about. Like our biostats department actually has like sort of happy hours and like help hours where you can go. And there's like 4 statisticians there. You bring them the question that you're trying to address and this potential stats you may want to do. And they will tell you or give you advice and guidance. And often then that starts a collaboration.
Very good.
Yeah, I guess I would throw in, we're talking a lot, we kind of spend a lot of the time talking about things from the perspective of someone writing or putting together a paper. I would say from the perspective of someone reading a paper, again, obviously you're not going to, unless you really, really care about the article, you're not going to pick it apart and try to do a statistical analysis yourself. But I think that there are some sort of thumbnail things that you can do to see whether what you're reading makes sense or not. And and to me, I think that I do tend to look at what they do their analysis on, and I tend to look at what kind of, usually we're talking about p-values or something of that sort. I tend to look at how many things that they're reporting have a significant p-value, and how many things I would sort of expect, based on the study they've done. If I was doing this study, what would I be looking for? And one of the things I try to look for is whether what they're reporting and what has good p-values corresponds to kind of what I would expect or what I would be looking to generate if I had done a similar project. And so I think that, that can give you a good idea. If it more or less seems like the results that they're getting make sense, and they're getting good p-values on the things that you would be looking for yourself out of a similar study, then I think that, you probably are dealing with a paper where, they're getting robust results and the statistics are fairly straightforward. On the other hand, if you have a situation where the p-values are, they're only reporting a few, or they're reporting things that seem sort of tangential, and and they maybe are not reporting things that you would expect them to report, then that makes me kind of wonder, and then you start scratching your head and thinking, maybe this isn't as robust, and maybe they did a whole lot of different analyses and they're cherry picking the ones that have a good p-value, but they're not really focusing on what should be the most important findings. And that's something that you might question and say, well, maybe this study isn't as strong as it looks on first glance. So, taking it from that perspective as a reader or reviewer, I think that those are some kind of just quick rules of thumb you can use to get an idea about whether the statistics seem, to pass the smell test, so to speak, just on a quick first glance. Yeah.
Very good. Actually, that's very helpful. And thank you for the suggested readings. That I think a lot of people would find that useful. I know I would. So now let's move into the discussion section, which again, at the end, all of the parts that we talked about, they merge in to bring the points home and capture the essence of the paper in the discussion. So I will open the floor to you guys, and let's see what you think is the best way to talk in the discussion. What would be what makes a great discussion portion of a paper?
Well, I think we need a little Shark Tank jazz hands, like the introduction. The discussion is the opportunity to actually then interpret the data, speculate a little bit on what that means, not overstate it, but you have a little bit more freedom there. And then, as Mike can, wax and wane eloquently with his beautiful English background, and, Vivian with the Shark Tank training, this is again, an opportunity to not lose the interest of the reader, but also to get your point across. So it's like, what do you want the reader to take away from your study? And the discussion really is where you can kind of hammer that home. So Vivian, I'll hand it off to you. What do you think about the discussion?
Oh, I completely agree. This is where you wrap everything up and you say, these are the reasons why this work is great. These are some future directions and a little bit of potential speculation. And then don't forget to put your limitations in there. I think you know your limitations best if you're writing this. And if you're reviewing, if the limitations aren't clearly spelled out, they should be, and you should ask for those. Because everyone needs to know that there are limitations of every study. And if we all as a group clearly state those every time, then that helps strengthen our research as a community.
Right. I would agree with it. I think that one of the biggest mistakes that people tend to make in their discussion is that, and again, this makes sense. I mean, they've done a lot of work on this thing. They've done all this work to do the research, get the IRB approval, actually write the paper for, it's many hours, it's a labor of love. It's almost like, I think Guli said in our practice, it's like your child that you're delivering into the world. And so, people have a tendency to want to make it seem more grandiose than it is, which is totally understandable. And to some degree, acceptable, just like parents who brag about their children, we kind of accept that. But at the same time, including the weaknesses and including things that, things that could be done better under different circumstances really helps. And it helps to ground you as an author, and it helps to reassure the reader that you are still kind of in touch with reality, and you're not saying things that are completely out of the realm of what's reasonable, and you can actually draw credibly from the data that you're presenting. And so, the biggest mistake people make is that they have very limited data, and maybe the study is limited for good reasons. They've done the best that they can, but then they try to make huge claims that, they've revolutionized this or that field when it's weak data, limited by for whatever reason. And really, they're just making one small contribution that, yes, it may be a good contribution, something helpful, something that does push the field forward, but usually not to anywhere near the degree that they try to claim. And so, as an author, when you claim too much, you actually turn readers off, whereas if you're more modest and you admit the limitations and you say, okay, I know that my study isn't perfect and these are the reasons why, that actually gives you more credibility. And whatever you have really discovered and produced is more likely to actually be accepted, and people are more likely to try to build on it in the future if they can trust you to, be more realistic about what you really have put out there. So, I think that as a reader too, you need to be always very cautious about, in the abstract or in the discussion, when people are claiming that they've discovered something really revolutionary, you always have to be, sort of skeptical about that and take it with a grain of salt and really look carefully at what they've done and what their data really supports before you start making big decisions or start, doing a big research project of your own based on something that may not be as sturdy and reliable as what the author is presenting it as. So that would be what I would add to that.
Very good. That's perfect. So now I guess we can start the fun, because the whole point of the discussion was how to critique a paper. I think it has been said in so many different ways during our discussions up to now, but I'm just going to make it a little mix and match. So I'm going to start with Vivian. So what makes a good reviewer? So in your role as the PI of an independently funded lab, and among many other roles that you have, you may be able to share with us some unique views that you would have as a critical reviewer of the literature and also as being reviewed in the work that you're doing. So if you can share with us, that would be great.
Awesome. Yes, I think reviewers are essential to our field. I think they hold us accountable, and I think they also strengthen the work that we do. And so as a reviewer of a paper, my job is to make sure that the science is solid and that the conclusions that are drawn are appropriate. And as a researcher, my job is to do the same, right? To make sure my science is solid and my conclusions are appropriate. And on the research side, when I get a critique, the most helpful thing for me is to see a critical review and ways to strengthen. And so then when I review papers, I try to give back the same thing, right? Just saying, well, this study isn't novel. That's not a helpful review, right? That doesn't help strengthen the field. It's better to give suggestions as to how to present the science so that we highlight the novelty of it. Or what other population can you bring into your study, or what else can you do to make it novel, right? So not only do you provide a critique, but you also provide recommendations to strengthen the work. And that's really essential. This is what I do. I mean, I research for most of my time, right? So when I present a manuscript, it is, as has already been mentioned, like the birth of a child, right? It's something we've worked on for so long. And so we really want the reviewers to give it that sort of respect and really help strengthen it to the best of their ability. As Paul mentioned before, getting a statistical review can be very, very helpful. And we have had reviewers recommend that, and it can be really amazingly insightful, even when we have statisticians already that are collaborating with us. So I guess my most important point is that you really should give a very critical review, but also suggestions for strengthening the work.
Perfect. That sounds very right. I mean, that's perfectly said. So now let's talk to Mike. So with your extensive involvement in many scientific and educational committees in ASC and other professional organizations, I'm going to ask a question that is kind of somewhat obvious, but I think it's good to hear it and to decipher it. So why do we review papers? So I'm sure you have plenty of reasons and you can share that with us. beyond what normally we say, okay, we'll review papers for this, but okay.
Yeah, I tend to be one of the people who does the most reviews for some of the articles that I do, or from the journals that I do review articles for. I guess to me, it is one of those things that's is sort of academic grunt work where you don't get a lot of credit for it. And as Vivian really eloquently said, if you're doing a good job as a reviewer, you're actually putting a lot of work in it to try to make the paper better, even though your name will never be on that. But a good reviewer can really, help the author to achieve more success by pointing out flaws or by pointing out ways that they could make their article more appealing and, more impactful for readers. So why do I do it? Well, like a lot of things in academia, I personally do find it to be somewhat gratifying to, contribute something and help people to make their papers better and help journals to produce a better product that, is more educational and has a wider reach. So, even though I don't get a lot of personal credit, I feel the credit internally, I guess, when I do it. And also to be honest, a lot of it is, somebody asked me a favor, so I say, okay. So it's a mix of just, trying to help people out and trying to help out the people you don't know, the people who are writing the article too, and trying to help out the readers so that when the article gets published, the readers have a more enjoyable experience as well. So, these are all the reasons why I find it gratifying. Now, why does it have to be done? Pulling a step back, I think Vivian already talked about this, that, the review does strengthen the article, and it should. Sometimes you get reviews back saying, oh, this article is fine the way it is. And that's actually, I kind of feel insulted when it's nice that they give it a pass, but it's actually kind of insulting in that they didn't put any effort into trying to improve it at all. The reason we do it is to, first of all, make sure that it actually is a credible work that, is not obviously wrong. Now, most of the time, we're not going to do a super deep analysis and actually go inspect their lab and see what they really did and do all that. But, we are there to make sure that it is at least a credible, reasonable result. And we also are there to make sure that they are producing something that will be valuable, to the community that they're producing it for. And so, it's not just a matter of, sometimes it turns into kind of a game of just filling pages in journals, unfortunately, because there are a lot of journals out there and they all need content. But, we are there. The real purpose ultimately is to make sure that what goes into journals is deserving of being there and deserving of being read. And if people do read it and take it seriously and do what the articles suggest they could do, that, it will actually make things better and not worse. So, that is ultimately the responsibility when you're reviewing a paper, to try to make sure that this article is going to make the world a better place.
Very good.
Boil it down.
Very noble cause. Very noble. Very good. Yeah. So now let's go to Paul. So we talked about the do's of the critiquing of the paper and reviewing a paper. So as the associate editor of JASC, among many other editorial roles, and as well as many research and educational endeavors, you are in a great position to share with us the don'ts of when reviewing a paper.
Wow. I will answer that question. I'll get to it. And I think that Vivian and Michael might also have some really useful thoughts there as well. But I'm going to take this opportunity, to maybe pull back the curtain a little bit for the ASC community, especially with JASC, as an associate editor. But basically, the way that we do things at JASC is very similar to most of the other, editorial boards that I'm on. And I think this might actually help, certainly the trainees, but also, more junior cytopathologists and pathologists to understand the process, like what goes into, getting, how does a bill become a law? How does your manuscript become a PDF in JASC? And so, when you submit your work to JASC, we'll use as an example. So at JASC, first goes to the editor-in-chief. So she looks at that manuscript, and if it is terrible, not a good fit, and not even worthy of review, she might do a desk reject right there and honestly not waste everyone's time, because a lot of time and effort goes into reviewing these manuscripts. So that's the first kind of checkpoint. If it looks like it's a decent manuscript, then what she does is she assigns it to one of the associate editors. So either myself or Martha Pittman at MGH, or Leon Pantanowitz at U of M are the three associate editors for JASC. And it's based on the topic of the paper, the expertise, how many manuscripts we have in the queue. And so once the associate editor gets that paper, what we do is, I read it thoroughly. Again, if in my opinion, there's just no value, it's a really terrible manuscript, it's not ready for review, I'll do a desk reject as well. Again, not to waste the author's time, because nothing is worse than having your paper languish in review for 3 months, right? And you're wondering what's going on. Whereas if it's going to be a reject, let's get it over with so we can either choose a different journal or fix it. So then after I read the paper, it's something that needs to be reviewed. Then I have to choose reviewers. So I have to send out invitations to people in the field, experts on the topic that the paper is about, people who have published about it before, people who are very good reviewers. So there's a large database that we can search to look and find out who might be the most appropriate reviewer. And so we send that invitation out, an invite, will you review this manuscript? And as Mike said, it is a favor, and it is a lot of work. Every time I get a review request, I take it seriously. I don't want to send back just a one-liner. We'll talk about that at the end, but you're going to take, half an hour at least, if not an hour or two, to give a really in-depth review. So then we usually send that out, and most papers have 2 reviewers at least, sometimes more when we get the reviews back, if they're conflicting, if one says yes, one says no, if they're conflicting, or if it's a really hot topic or an important paper, we might want a number of opinions on that. And so we send them out. A reviewer will accept it. They'll review the paper. Usually we ask the review to be sent back in about 14, 15 days, so a relatively rapid turnaround time, understanding again that you as the author want to get that information back as quickly as possible.
Right.
So after the reviews come back, then me as the assigned associate editor, I read the comments to the authors, I read the comments to the editor, and then I make a decision. Is it an accept? Is it revised with minor revisions? Is it a major revisions, and if they're done, we'll reconsider the paper? Or is it a flat out reject? And so we try to put it in a box, and again, take all of that information that we have been given. And so the reviews are incredibly important and incredibly helpful because you want a lot of viewpoints on that paper. Oftentimes the editor themselves will also add other comments or things that the author should address. And then that goes back to the editor-in-chief. She, Dina Modi, then sends out the letter, either of acceptance or revisions are needed, goes back to the authors. And then if revisions are needed, then it comes back to us and we do that process again until we get to accept. So it really is an iterative process. It's an in-depth process, and a lot of work goes into it. And as Vivian and Michael both said, a good review and was really helpful as to the editor are frank comments. What are the strengths? What are the weaknesses? What needs to be addressed? And then to the authors, how can you improve that study? I think that we've all danced around this. We all want to make papers better. We want good things to be, put out into the world. And so this is something in academics that we do. Every paper that I review, I learn a little something from, and you get better at writing papers from reviewing. So that's something that you can benefit from. But we really do want to, in JASC, in every, journal that I'm associated with, the review process always strengthens a paper. And personally, even though it can be soul crushing to get that rejection or that really harsh, or as Guli's, mentioned in the chat, the dreaded reviewer number 2, comments, every time the paper always comes back better, even if you can't see it in your own paper after the first one. So I spoke a lot. I'm gonna toss it to Vivian, Mike. Any other thoughts about that, or don'ts of what not to do in a review?
I think, a good example of the importance of the review process has been actually, in the last year during the pandemic and all the research that has come out about COVID-19. We have seen major journals publish really important works on COVID-19, some of which had to be, retracted and withdrawn, and the results to be were found to be invalid. And actually at Vanderbilt, we did a very in-depth journal club for some of the graduate students that I work with, looking at some of those articles that were retracted and that were found to be inappropriate in a number of different ways, right? And so that really highlights the importance of reviewers, particularly with really novel, cutting-edge research that's done quickly, right? I mean, we all know that there's exciting, groundbreaking research that's done that is pushed through at an important but fast pace. And so that really highlights the importance of the reviewers to hold people accountable, to look carefully at the statistics, to get that statistical review if it's needed, and to really think critically, not just write the one line, this is great, so-and-so is a great researcher, we know them, we're going to say this is really great, but critically spend the time to review the article.
Very good. Anything, Mike?
I would throw in one last quick thing. One thing that I always do when I'm writing a review is that I don't push the send button right away. I let it sit at least for half an hour, go do something else, check my emails or sign out a few cases or something, and then come back to it and read it again. Make sure that, there isn't anything in there that seems unduly harsh or might be interpreted as being overly negative. So that's just one, to try to make sure, because it is tough as a reviewer. You're giving feedback, and usually most of the feedback is negative. And some of the papers just aren't very good. And you do sometimes get frustrated in the process of reviewing as you see so many obvious flaws that you feel like you shouldn't have to point out to them. And so sometimes you, as a reviewer, do sometimes maybe get overly harsh. And I know I am guilty of that sometimes. So I always try to do a harsh check before I push the send button and try to mellow it out a little bit so that it doesn't come across too harsh. That's something I've learned. So I would give that advice to anyone who's thinking about doing reviews.
Yeah, and Katie, so then I didn't completely ignore your initial question. So I have two don'ts. So two don'ts as reviewers. And number one is exactly as what Mike said. Don't be overly harsh or cruel or mean or vindictive. Even if it is a terrible paper, there is some good in there, and a lot of hard work and blood, sweat, and tears went into that paper. And so constructive criticism, how can we make this better? Even if there's no way that this is ever going to get into your journal, how can you help the authors so that they can reframe it, they can find a better journal, they can actually make it a better study elsewhere? So number one, don't be mean. Don't be cruel. Make the world a better place. Haha. All that. Second thing is, don't, and this is something that it can be very difficult, and for myself as well. And talking about how to present the data and the tables versus graphs and all of that, I have to find myself sometimes teasing apart what changes need to be done to the manuscript or the way that that is presented, et cetera, versus how I would do it. And just because it's not the way that I would do it doesn't mean it's wrong or that other readers might find it fine. And so I find that balance between, sometimes I'll offer suggestions like the authors could consider doing XYZ, or they could consider reframing or, reframing the study this way. That's not like an absolute, they must do this. And some of those comments are take it or leave it comments. And I think those are helpful to the authors. But just because you really firmly believe there should not be 7 tables in every single paper doesn't necessarily mean that a paper is not good if there is 7 boring
tables. Very good. Very good. Thank you so much for giving the answer to questions. I think at this point, we can have a QA session. And I'm going to ask Dr. Barkan if she can actually, because I'm having technical difficulties today. Now I can't even scroll through the questions. And so if I can have phone a friend to have help with that, that would be great.
Anytime you can phone a friend.
Thank you. Thank you.
Let's see. Let me ask one of my own questions for you guys. Do you use reference managers when you're writing papers? And if so, which one do you like using? Paul's laughing.
Well, I'm going to jump in here because I'm going to out myself just as a total Luddite. So I do everything by hand. I don't use any reference managers. No paper I've ever written have I used RefWorks or EndNote or anything like that. And perhaps it was because, so back when I was a grad student, my PI, when she was writing her, my co-PI, when she was writing her dissertation, EndNote crashed. And so she was like, I'm never doing that. And so I learned just to do everything by hand. And honestly, I've got a great system, and people look at me crazy. I see Vivian's eyes are wide, like, how could you not use EndNote? But it forces me to have everything in my head. And I've got this great system where I write the paper, I do the revisions, other people will look at it. And then it takes me about 15 minutes at the very end, right before I'm ready to submit, to change all of those kind of parentheses, paper and all that into the ordered thing at the end. And so that works with me. I'm a terrible Luddite. I still, pen and paper, in my pockets. If I still could have a Razor, cell phone and not a smartphone, I would. But so that's what I do. But now let's see what actual real, scientists and pathologists do.
Well, I think actually that's really great. Full disclosure, I use EndNote and I don't know what I would do without it. Okay. I could not do what Paul does, but it actually is a really smart idea. And one reason is because as you put references in and stuff, and then you get the numbers and it formats them for you at the bottom, it's very easy to put in the wrong reference. It's really easy as it reformats and renumbers the whole thing as you edit and delete for the wrong references to get in there. You hope it's doing it correctly, right? I rely on it tremendously, but if you don't do them by hand, it's important when you're all done to go back one by one and check all those references. And Paul just does it automatically because he waits to the end and formats them beautifully. But even if you use a reference manager, that's a critical step because readers will go to those references. They want to learn more about that topic or they want to see that data. And so you're doing them a disservice if you haven't checked those references.
Mike, how about you?
I was going to say the same. I use EndNote, but it's flawed and it has to be carefully checked. And I see that's actually one of the pet peeves I have reviewing papers or even reading papers that are published is that so often there's obvious errors in the way the references are presented. Mm-hmm. And, and I understand why. Oftentimes it's very obvious that it just goes back to the algorithm that EndNote uses that's screwed up in some way. But, it just shows, these authors, they put so much effort into this, and then they don't follow through on the, just the basic double checks at the end. And it makes you wonder, how serious they really are about this, And so, and as has been said, you really want those references to be correct because oftentimes that's what people want from your paper as much as the content of your paper is all the references you already found so that they can learn more about the topic quickly without having to do their own search. So the references are really important. And, not just making sure that you're thorough, but also making sure that they're accurate and that people can actually follow them and find the articles correctly.
Yeah, totally second that. I am a believer of EndNote too. Without it, I don't know what I would do because I am not very good typing, Paul, like you. So that would be an issue for me. And as a reviewer, when I look up, papers, I do kind of spot check some of the references just to make sure it's in the same ballpark. Like if it's about urine, there's no mesothelioma thing in there by mistake that got in. But I'm admittedly, I don't check every single thing when I'm just reviewing a paper, but when you're sending it, I think that's a good idea. Now here's a question from one of our audience members, and they say, is there a software that you recommend for data presentation? I suppose other than Excel, tables or graphs, or I don't know, is there anything else?
There's one that and I may be behind the curve here, maybe people know about this and have known for a while, but there's a software called biorender, which allows you to put together your own diagrams. It's not going to analyze the data for you, but if you need an explanatory figure on how something works, or maybe a figure, even for the supplement, as Paul suggested, to detail your methods, it has beautiful, already pre-done graphics that you just, I'm not super computer savvy, but I can very easily use biorender to make some graphics. So that's a new one that I wasn't familiar with until recently.
Good to know. Any other programs that you guys know or like or recommend?
I can biorender is awesome. Again, if you're making little, cell pictures or, mechanisms, that's really useful. Personally, I use mainly, Excel and PowerPoint. And I think that in newer iterations, just the functionality and the aesthetics have really taken a notch up. And so there's a lot in there with the basic programs of PowerPoint and Excel that you can make some beautiful charts, graphs, etc. It's always good. Just again, a little editorial note: you don't always have to use the default colors. The green, red, purple. Like everyone knows what PowerPoint will automatically spit out. And there's also plugins. You can buy plugins for Excel and PowerPoint for pure graphics. And then there are other statistical programs that have company graphic programs as well. And then once again, I have a lot of collaborators with MPHs and other researchers that have other either homegrown or other programs as well. And so, great question. And so, but again, personally for me, Excel and PowerPoint can get you 80% of the way there.
That's good. Okay. And this is probably as good as time as any to remind everyone that actually a picture's worth 1,000 words. Instead of having lots of prose and tables, if you have one beautiful picture, then that, conveys the message and people understand it so much easier.
Right.
And
Yes.
Right? Yeah. Nicer. Let's see. There is a question, and let me see if I can rephrase the question. In terms of statistics and the use of statistics, and this is probably an area that majority of us have issues because none of us really are statistically based. But when do you decide to write things in a descriptive manner or use statistics? And what is the approach to decide descriptive versus actual statistics analyzed in data in the papers?
I guess I'm not entirely sure what do they mean, like in a morphology study, describing whether.
I'm assuming that this is more of a morphology study.
Right. So, whether you should actually do some kind of a statistical analysis of what different pathologists thought in terms of the different morphological aspects, or whether it's okay to just give a description of what different people saw. Is that kind of what the question is getting at?
I'm trying to understand the question myself, but I think what it is that, say you're looking at melanomas in, say, FinPac.
Yeah.
And do they have intranuclear inclusions? Well, yeah, 200 cases, maybe so many X of them had the intranuclear inclusion. And does it mean, do you do a statistics based on that? And this is a tough issue because, again, I don't think many of us during residency have a statistics training. And, those of you who are lucky to have a statistician on board on staff, that is great to go to. Some of us don't have that. And then what do you do in situations like that? I think that's the issue. And I kind of wish sometimes the journals actually kind of provided help in that, where you could say, I have this paper, I wrote this much, but I need help. What do I need?
Yeah, so I can jump in there a little bit. Unless if you're writing an editorial or maybe a review article, if there are no statistics in your paper, it's probably not going to get accepted. You really, if you're making some comparison, if you're comparing means, if you are, if you want to say, 60% of AUS FNAs have grooves or something like that. Well, what are you comparing it to? And if you're in a percentage, a number in isolation is not very useful. So you really do need some statistics. And again, very simple ones like a chi-square or, a t-test, a student's t-test, just to compare means. If you're having a control group and an experimental group, you do need to have some statistics. And yeah, some papers come through where, again, they're describing 80% of this group had XYZ and only 20%. And it seems like, wow, that's pretty far apart. And maybe that is useful diagnostically, but you still need statistics. And even if it's a small group, you do have to have some statistics is kind of the take home point I'll say there.
Yeah, I was going to say, it's sort of like the difference between a case report and a study. Once you get past 2 or 3, then it becomes a study and not just a case report anymore. And so once you're trying to publish something as a study, you do need some statistics. And for the most part, in a lot of these, I think people get intimidated because they see, oh, there's so many things I could do statistics on, and it's so difficult. But actually, once you figure out the first one, a lot of times most of the others, you just plug the same formula again. So it's really not as intimidating as it seems. And that's been my experience working on these kinds of papers. I get intimidated, think, oh boy, I have to calculate 50 different p-values. But actually, after I get the first couple done and I feel confident what I'm doing for this particular study, then the rest is kind of roll off, and it's not a big deal. So,
agreed. Even the most qualitative studies could actually use some statistics, which kind of give it power and make it more understandable and put things into perspective. Here's another question from one of our participants, and they say, okay, you talked about tables and pie charts for presenting results. What other ways do you suggest results should be presented?
Yeah, great question. Sometimes text is good. If you don't need to create a table. If in your table, you're just listing 3 bullet points, or you're just having, or if in your table, you only have 1 column, tables are useful if you have, again, multiple columns and whatnot. But sometimes if it's just short, or if you're only talking about 5 different things, maybe it's better just in your results to say, condition 1, X%, 5, condition 2, condition 3, condition 4, and not create a table. So I'm going to put text, just text is very appropriate. And then just the whole, there's so many different charts and ways to visualize data. It's exciting to when you just, okay, so kind of geek out here. When you do stumble across a new way of presenting data, a new chart, something that you haven't used before, like that gets me really excited. And I just can't wait to put something like that out into the world. And when you have that perfect marriage of a unique, new chart or way of visualizing the data with a data set, as Mike said, then you have your killer figure that is going to be, people on the Twitter machine at conferences will be taking, pictures, during plenary sessions and whatnot of your figure. And then it got retweeted and whatnot on social media. Again, what I hear. But yeah. So that's what I have. I don't know, Mike, Vivian, any other thoughts that are more intelligent than what I just babbled on about?
That was very intelligent, Paul.
I think bar charts are overused in science in general. Bar charts are an easy go-to, and I use them, definitely. But if you've got 6 bar charts in there, maybe you should start thinking about another way to show the data. Because just like tables, you can get lost in bar charts.
That is true. Paul, that's interesting. You made me think the first time I learned about funnel plots was from Dr. Pambucha, and he taught me how to do that. And it was brilliant. I'm like, oh, this is such an interesting way. I've never seen it in any of the pathology articles before. And he just quickly put it together. And I'm like, oh, this is great. So I share your enthusiasm with these new ways of showing things.
Yeah.
Let's see, there's another question here, and that's a very good question, and more on the kind of the editorial part, I would say. How much grammatical editing is the responsibility of the journal, like the words, spelling, phrases? If language is compromised, but the work is good, how do you handle it? Sometimes you get these reports from reviewer 2, typically, who go over the entire manuscript and point out your spelling mistakes, which you shouldn't have done in the first place, but it's there. Is this what we should be doing as a reviewer?
Sure, I can mention that. It's always helpful if when those really fastidious reviewers have the major comments and then the minor comments, and the minor comments is like line 38, you missed a period. Line 14, this should be this word, not this word. All of those errors. I don't think that, again, personally, it's great if reviewers want to do that. I don't think that that's specifically the job of the reviewer. I think that just pointing out there are a number of typos and spelling errors, or the English language is a bit rough, it needs to be, fixed for a, major revisions or minor revisions. So that's one thing. Number two, when a paper is accepted, there is on the journal and by the publisher itself, there is a round of some smoothing over and some cleaning up of the language, of some typos, before the proofs are sent out to the author. And then the authors will review the proofs just to make sure everything is okay. And my third point is that at the end of the day, really the responsibility lies with the authors. It's very embarrassing to put a paper out into the world with spelling errors or with numerical errors in one of your tables, or if something is wrong. That reflects poorly on you as a scientist, as a researcher, as an academician. So so there are some checkpoints along the way from the journal's perspective, but the onus ultimately relies on the authors.
And as a reviewer, is it okay to say maybe an English review is warranted in a kind and gentle way, not in a mean kind of way, correct? Yeah.
Yeah, absolutely.
Yeah. There are any number of papers that the main problem is just that it's not very well written, and it's not, somebody who English is not their first language, and they didn't translate it well. Mm-hmm. And it just makes sense as professional services that are easy to access, that people can send these things to help them to make the English better. So, I think there's nothing wrong with, as you say, gently suggesting, and in some extreme cases, even requiring that they do something like that.
Yes. And I think I could say this as a person whose first language is not English. It is nicely to put it nicely, like, English language review is a good idea, but you shouldn't also be insulting to the author by saying, or their first language wasn't English or something like that. That is not a kind and gentle and appropriate way of doing that. Then I'll give you.
As Paul said, we should definitely be kind. And yes. I think Mike mentioned this as well. It's really easy to be overly critical. You're having a long day, you're reading quickly, you're frustrated, you're trying to get it submitted, and you just spout out and text what you were thinking about the article. And as Mike mentioned, reading over and making sure that you're really being thoughtful and kind with your wording is so important.
And so with that, I'll throw out a little, just a little anecdote. As a graduate student, my first scientific paper, I still have this nightmare, but I remember, I worked on this adaptive transfer model of NKT cells, looking at atherosclerosis and lipid metabolism. 2 years of work, submitted it to the American Journal of Pathology. So excited. Got the reviews back, and they were terrible. It was a flat out reject, and it was just brutal. And I remember just feeling absolutely crushed. And like I was thinking, what's the point of this? Why are people so mean? Why, I work so hard on this. But then, kind of as how do you deal with? So why do I say all this? Number one, to show that we're all human, and we all get papers rejected. And when you get the harsh review or the critical review, how do I deal with that? So what I do is I read it, and then I let it set. I don't react. I don't shoot out an angry email. I don't, do anything. I let it sit for a day. I go back to it with a little bit more of a level head. And it's like, all right, there really are some pretty good points that the reviewers brought up, or man, I really should do these experiments. And, I didn't think about that. Let me do that. And so, again, the whole goal of the peer review process is to make the paper better. Sometimes you run into, again, GUIs, the evil reviewer number 2. And so sometimes you just have to cut bait with that journal and maybe try another journal and try another reviewer. But in general, reviewers do help. You can work the system, you can resubmit, you can show that you're making those changes. And then even with that paper, with a flat out reject, it was accepted eventually. And so there is a happy ending at the end of that story.
Nice.
With small little, graduate student Paul, crying in the corner that his first paper was just obliterated.
Yeah, I would say that too. The one thing that often as reviewers we tend to forget is that a lot of these things, the first person who reads it and the person who really cares is a trainee or a student who doesn't have experience with these things, and they often are very fragile, actually. So that's another reason to try your best to be level-headed and not be too harsh, because you're thinking, I'm sending this to some 50-year-old guy who's already written 100 papers, but that that may be true of the senior author, but the person who really cares, the trainee, this may be the first paper they've ever written, and you should try to keep that in mind.
I love that. Empathy skills as a reviewer. You should write on that. I love this. This is so nice. Well, you guys, there are some more questions, but it's kind of the top of the hour. I want to slowly kind of wrap this up. So one of the things, and I'll tell you what I think, and then maybe, you let me know if there's any last words. But when I'm sort of reviewing a paper or, presenting at a journal club, one of the things I think to myself is, if I were to do this study myself, how would I change it? How would I have designed it, barring any time or, of course, monetary concerns and whatnot? And did they really answer the question that they set out for? So to me, that's one of the important things when you're critiquing a paper. How about you guys? Final points from you guys. What do you think? If you want to leave our participants with one or two thoughts, what do you think?
Well, I guess I can go first. I think that, as you said, empathy, my two key takeaway points from this kind of have to do with that. So the one thing to keep in mind with any article when you're writing it or reading it is that there's a huge sort of disconnect between the authors who have put a lot of work into it and who are very intimately familiar with what they're doing and who care a lot about what they're doing, presumably, versus the readers who, for the most part, probably care a lot less, know a lot less, and put a lot less effort into reading. And so that you have to bridge that disconnect with empathy on both sides. And, so when you're an author, you need to pull back a little bit and say, hey, wait a minute, how can I make this something that somebody who cares about this a lot less than me will still get excited about and think this is worthwhile and go all the way through to the end and read all of this and try to understand it. So you can't assume that everybody who might read your article cares about it or knows as much about it as you do, and you need to write it with that perspective. On the other hand, as a reader, you do need to, and we talked about this several times, as from the perspective of a reviewer, but just as a reader as well, you need to keep in mind that this is somebody's baby, and even though maybe there are some flaws and it's not the way you would have done it, as has been mentioned, there still usually is something worthwhile in there. And if you're reading it at all, it's probably because it has something to do with the topic that's of concern or interest or value to you. And so pretty much anything that gets published by a reputable journal, it's going to have something in there that's valuable and useful. So you should, it does take some work sometimes as a reader, maybe more than might ideally be the case, but there's usually some kind of nuggets of value you can find in there. And it does make sense to maybe put a little bit more effort into some of these things and not just skim it quickly and give up on it too quickly. So, but the other thing on the other hand is you need to be skeptical as a reader as well, because as I talked about before too, the authors pretty much always overstate the significance or the definitiveness of what they found. So you do need to also maintain your skepticism and say, well, does the data really support what they're claiming in the conclusions? And a lot of times it only half supports it. So you have to kind of keep all of that in mind. And like Guli said, having the empathy skills to understand the perspective of other people is key to, keeping all things in perspective and getting the most out of what you're doing.
Very good. Actually, we had some take-home points by the panelists that they put together very nicely. If Joanne can put the presentation on, because again, somehow I'm not connected to anything right now. I'll just see you guys and hear, which is great, but I guess I don't know.
Well, I did mine. I just did mine. Oh, here it is.
Yes,
okay.
You kind of did.
No, I did mine verbally, but yes. We can go. Yes. Okay.
So I guess this is Vivian. So
these are mine. I think this focuses on the introduction, but just as a whole, tell a really good story. The introduction is your first impression. Lay out your points nicely. The rest of it should be, how you're telling your story and then how you wrap up your story at the end. And so if you follow that Shark Tank model of really telling a good story, you're going to get more readers and you're going to advance the field.
Very good. Mike already, I'm trying to.
I think this is covered. We can move on.
Yes, your balanced approach was very well said. And I think we're moving on to the structure of a good manuscript review by Paul.
Yeah. Again, a little bit self-serving here, but when you are asked to review a paper, there are two main, regardless of the journal, there's the comments to the authors and there's the comments to the editors. And the comments to the authors, especially for the trainees or those who haven't done very many, is there some sort of an outline that you can follow? So I think that the way that I approach it, and I think a good way to kind of structure it is in the comments to the authors, briefly summarize the paper in 1 or 2 sentences, just to prove that A, you read it, and B, you understood it. Next, say, what's good about the paper? Is it a hot topic? Is it a novel idea? Why should the readership care about it? What is the core goodness, or what does this paper contribute? And then go into that list of the points that the author should address or correct. And this is really most of the review, and it could just be a few minor points to, 10 major things that the authors need to address. And so kind of having that type of a pattern, something very, very reproducible and something that's very useful to the authors. And then if you go to the second, slide, is the confidential comments to the editor.
I'm trying to do that.
They're confidential.
There we go.
I guess, yeah.
Excellent. And this is, again, from an editor standpoint, again, the everything you type there is only going to the editors. So the authors are not going to be seeing this. And really, what's the purpose of this? Some people just copy and paste what they said to the authors in the confidential comments to the editor. But here, I just want to emphasize, this is an opportunity for you to have more of an informal kind of conversation with the editorial board. So you get to share your gestalts. Is this a good paper? Or is it a bad paper? Is it timely? Is this a diamond in the rough, or are there fundamental problems? You could be like, I really think this needs to be accepted, but the authors absolutely have to address points 2 and 4. The others are take it or leave it. And so this is, again, just really good feedback for the editors, so that we don't have to just read between the lines of what you're saying in a very kind, empathetic, in a very, empathy-laced way to the authors, as we have talked about, but this can be very frank. They tried really hard, but this is a train wreck. I don't see this being rescued. You don't have to tell that to the authors, but you should tell that if that's what your gut feeling is, to the editors, because again, we want to have an iron fist and a velvet glove, right? We want to be very gentle and kind, but we're not going to publish bad studies. We're not going to put bad things out into the world. And so, we have to be gatekeepers. And so the confidential comments to the editor is a really useful tool that I think is very underutilized in most reviews that I've seen. So, so those are the points that I wanted to share.
Very good. Thank you so much for all you guys have done. And I, for one, learned a lot. And I appreciated all the help that the last minute push came to this. So I believe before we say goodbye to our audience, and I know that we went over, but there is a poll for the question that on a scale of 1 to 5, how helpful was this session in improving your understanding and how to critique a paper? 1 being the least, 5 being the most helpful. And okay, that's
very nice.
Very good. That made my day, I believe, and I again thank everybody for their attendance and participation. And I will just put a plug in for the March 17th, 2021 for another interactive eJournal presentation, Learning Cytology in Times of Pandemic, and that couldn't be timelier, I believe. So hopefully to see everyone on March 17th. And with that, I think we can say goodbye and thanking again, everybody.
Thank you all very much for doing this. This was a wonderful session, and it's a nice kickoff to the eJournal committee. But it's also next week, don't forget, we have another session. We'll be talking about quality assurance issues. Till then,
Very good.
See you all.
Thank you very much.
Thanks, yeah.
Thanks, everyone. It's a lot of fun.
Thanks. Very good.